Welcome back to Biochemistry on Catalyst University. My name is Kevin Tokoff. Make sure to like this video and subscribe to the channel for future videos and notifications. Alright, so in this video and the next one, we're going to be talking about two pathways that are processes that are mostly done by the skeletal muscles. It's a concept that's very common to talk about in an exercise physiology kind of course. And the Cori cycle and the glucose-alanine cycle. And they're very similar in their functions, and they're often confused. I know I confused it a lot when I first learned about them. Both of them are going to involve the use of this enzyme, which is very important to understand. It's called lactate dehydrogenase. Now, without even worrying about the structure so much, this structure on the right side, pyruvate, this is, of course, the end product of glycolysis, pyruvate. Okay? It turns out that under more anaerobic conditions, when we don't have very much oxygen present, the pyruvate will run to the left and be converted to lactate. And this is catalyzed by a reversible enzyme, lactate dehydrogenase. So under anaerobic conditions, pyruvate runs to the left and we get lactate. Under more aerobic conditions, any lactate that we have tends to run to the right by the same enzyme because it's reversible into pyruvate. Okay? And we're not going to go into the implications of why this reaction is so important. But one thing we do want to focus on is how both lactate and pyruvate in different ways can be used as a fuel source for skeletal muscle, particularly during exercise. So now what we're going to do is focus on the Cori cycle. So here's the Cori cycle. Now this on the left is the liver and renal cortex. I really just want to focus on the liver because the liver is the main one that does this. And over here on the right, it says muscles, erythrocytes, which are red blood cells, and so forth. I want to focus on skeletal muscle because that's really what we're mostly concerned with. Skeletal muscle are the workhorses of the body. Okay? So let's start off in muscle. This is a good place over here to start. We have glucose. And we know that glucose goes through glycolysis, the process of splitting glucose, into pyruvate. Okay? We end up generating two NADHs, okay, whenever we break down glucose. We also generate two ATP, which is really important for the working muscle because myosin proteins have to use ATP. We have two pyruvate, but if we're under anaerobic conditions, which is what we've been talking about, anaerobic conditions, we can't actually use the NADH for anything. NADH only really works for the electron transport chain when it's under aerobic conditions. But for an actively and vigorously exercising skeletal muscle, NADH cannot be used in that way. So we're going to have to get rid of the NADH in some way. So what we're going to do is we're going to put the electrons from NADH on pyruvate and make lactate. Okay, this is catalyzed by LDH, the enzyme we just saw, lactate dehydrogenase. And we get two lactates. All right, we can talk about this NAD that's made through this process is reused in glycolysis. Okay. So in the muscle, we're generating ATP only through glycolysis because we're under anaerobic conditions, very heavy exercise, and we're generating two lactate. Now the lactate, if it continually built up in muscle, you would experience muscle soreness far faster than you do. Okay? You still experience soreness, possibly due to the lactate, but you would experience it a lot faster and a lot more strongly and acutely if this lactate was not transported out of the muscle. It turns out that that lactate, once it's made, a lot of it is dumped into the blood and it gets shunted to the liver. The liver will uptake that lactate. And in fact, the liver will now run the reverse reaction of lactate dehydrogenase, which is going to go from lactate to pyruvate. Because the liver is able to operate more on aerobic conditions. Okay, so the liver is going to reverse this. It's going to convert the lactate back to pyruvate, and then whereas over here we converted glucose to pyruvate through glycolysis, we're going to convert the pyruvate back to glucose through the process of gluconeogenesis. Okay? And that gluconeogenesis gives us more glucose. That glucose is not going to be used by the liver. The liver more is going to run on lipids itself. So the glucose will then be shunted back into the blood and be uptaken by skeletal muscle. Okay? And this occurs in a cycle, and it's called the Cori cycle after the Coris, Mr. and Mrs. Cori, that discovered it. Okay? And so what's the key thing from this pathway? Muscle cells use glucose. Through anaerobic metabolism, because we're probably doing high-intensity exercise, the glucose is going to be converted to lactate. The muscle cells will dump out the lactate, and that lactate will be picked up by the liver. then the liver just reverses everything. It turns the lactate back to pyruvate and the pyruvate back to glucose. Then the liver feeds the muscle cell glucose. In other words, the liver is taking the waste product of muscle, lactate, and converting it to a usable fuel source, glucose, for use by the muscle cell. So it's kind of a cool concept. The waste product of the muscle is the building block to make glucose, which is used by the muscle. And this just occurs in a cycle, and the transporter of glucose to the muscle and lactate to the liver is going to be the blood. Okay? And that is the Cori cycle.